Okay, so we'll start with presenting what are our goals for this lecture. So first of all, we want to understand what is spatial biology and why it is important. Second, we want to understand what is multiplex imaging and how it helps us to understand the tumor microenvironment, and what are the current limitations of multiplex imaging and how we can harness deep learning in order to overcome these limitations. So when I started doing this presentation, I started by doing what I think most of us do when they have a new project, is going to ChatGPT. And I asked him, so if he can explain simply what is spatial biology. And I think he had a really nice answer. So spatial biology is like looking at a puzzle and trying to understand how all the puzzle pieces fit together, instead of just seeing the pieces individually. We also pay attention to where they are in relation to each other. And this is what spatial biology is all about, is really to understand how the tissue organization and how cell and cell interaction really affects their function, and how we can utilize that to better understand diseases and to make better treatment decisions for patients. So what is spatial biology? Spatial biology is the study of biological systems in their spatial context and understanding how the organization affects the function. And actually, spatial biology started many years ago. We have H&E slides, which is the basic pathological slide for more than 100 of years. And we can even get some notion of the tissue organization from the H&E. We can identify tumor areas, we can identify tumor heterogeneity, which was used to grade tumors and still used to grade tumors. And we can also define some cellular subtypes. We can identify tumor cells, stromal cells, lymphocytes, but we can't really go into any more resolution or to gain another insights. Then immunohistochemistry was developed, and the immunohistochemistry allows us to stain for single proteins. And in that case, we can stain, for example, CD8, which identifies CD8 T cells, and then we can look at the architecture of the tissue and see where the CD8s are located. Are they infiltrating the tumor? Are they excluded? And we know that these properties have significance for the treatment outcomes of the patients, especially in immunotherapy. And now we reach a stage where we have newer analysis methods such as multiplex imaging. And now we can get into a much higher resolution. So we can have cellular subtypes, and we can have their cell state markers to understand really what are they doing, what is their function within the tissue. So now, for example, we can look and identify PD-L1 positive tumor cells, and to identify whether they have CD8 PD-1 positive T cells in their proximity. And this really allows us to enhance the biological insights that we can get from pathological samples. So in spatial biology, we're trying to utilize advanced imaging and analytical techniques to visualize and map the distribution of cells. And then we use this high resolution spatial data to uncover patterns, correlations, and spatial relationships within biological systems. So what are the current leading advanced biological methods? So the current leading imaging modalities are multiplex imaging, which is the use of fluorescent labels to visualize multiple proteins on the same slide. We have spatial mass cytometry, which instead of fluorescent label, it uses metal conjugated antibodies to image different proteins. We have spatial transcriptomics, which can be used to spatially to do spatial sequencing of the whole transcriptome in one slide. And we have also in situ hybridization based technologies, which allows us to map both RNA molecules and proteins also in a spatial context. And each one of these modalities has different advantages and limitations, but something that all of them has in common is their complex analysis. Because you get very high dimensional data, you get millions of data points in one slide. Each one of these data points has several dimensions, depends on the number of markers you can identify. And then the question is, how can we leverage these techniques in order to get the most significant biological insights and to really improve patients' outcomes? So why is spatial biology important? It has several very important functions. First of all is understand the disease mechanisms, so we can look at the tissue architecture and see, for example, tumor heterogeneity and how it affects treatment responses and how it affects proliferation of tumor cells, and we can see where are the immune cells and how they affect the tumor biology. And we can also allow us to really do better biomarker identification and to advance precision medicine. So right now we have a lot of treatment options for patients, and using spatial biology we can really focus them to the treatment that can most benefit them. And it can also help us enhance drug development, because now we can look and find interesting cell-to-cell interactions which might be druggable. Because spatial biology has immense importance in immunotherapy, and immunotherapy really revolutionized cancer treatment in the past decade, we can see an exponential rise in the number of publications relates both to spatial biology or to multiplex imaging or to the RNA-based modalities such as Visium, which is the whole transcriptome, which is spatial transcriptomic, and GeoMax of nanostring. Now, the reason that this rise is happening is because really spatial biology is very important to understand immunotherapy and to understand both response and resistance mechanisms to immunotherapy, because it provides crucial insights into the tumor microenvironment. In 2014, the first spatial biomarker was reported for PD-1 blockade, and in this case, they focused on the CD8 fraction within the tumor invasive margin, where responding patients had a lot more CD8 T cells within the invasive margin, and they also had a higher proximity between PD-1 and PD-L1 positive cells. And since then, there were other biomarkers that were suggested, but in a recent meta-analysis that was published in JAMA Oncology, it showed that multiplex imaging-based biomarkers were the most predictive to PD-1 blockade therapy outcome, and it was more predictive than IHC-based biomarkers, which are the companion diagnostic for PD-1 blockade. So right now, if by IHC a patient has high PD-L1, it can receive immunotherapy as first-line treatment, for example, in lung adenocarcinoma, in lung cancer. And multiplex imaging-based biomarkers were more predictive than these markers, also more predictive than DNA and RNA-based markers. And as our understanding of the tumor immunology grew, we also found more complex biomarkers. For example, tertiary lymphoid structures. Tertiary lymphoid structures are organized lymphoid-like structures that can form within the tumor microenvironment and are composed from B cells, T cells, and dendritic cells in a specific architecture. So now, in order to identify them, one marker is not enough. We'd need at least a B cell marker, a T cell marker, and a dendritic cell marker, and usually also a tumor marker to see whether the TLS is in proximity or not to the tumor cells. And several papers demonstrated that the presence of TLS is associated with a better outcome to immunotherapy. But actually, a recent paper that was published last year demonstrated that it's not enough to know whether there is a TLS or not. Actually, the cell composition within the TLS is also matters for the immunotherapy response. And they show that, for example, if you have high Tregs content within the TLS, your chances to respond to the treatment are lower. So now we need the markers to identify the TLS, but we also need markers to identify cells within the TLS. And the complexity just grows. So with our understanding of spatial biology that grows, we need more advanced imaging tools in order to be able to visualize more proteins simultaneously. And as our imaging tools advance, we can also our understanding of spatial biology grows. So it's kind of a reciprocal relationship between these two. And the main imaging modality we're going to focus in this lecture is multiplex imaging. And what is multiplex imaging? It relates to a group of imaging methods which enable staining of multiple protein markers on the same slide simultaneously. So now we have one slide. We can have simultaneously. We can stain for 50, even to 100 markers at the same time, and that really allows us to get better cell resolution. So, for example, if in H&E we can identify only lymphocytes, now in multiplex imaging we can find CD8 T cells, CD4 T cells, regulatory T cells, and their activation states, whether the CD8 T cells are positive for PD-1, for LAG3, for example, which affects their function greatly. And these imaging methods rely on the excitation and emission spectra of fluorophores, and thus they are really limited by the spectral overlap. So how it works is we have a laser that excites certain fluorophores, and then we have filters which can receive the light in the wavelength that the fluorophore emitted. And because we have a spectral overlap between fluorophores, we can usually stain without a significant spillover up to 5 to 6 fluorophores on the same slide. But if we want to go above that, we can't. And to overcome these limitations, most methods use cycling of staining and deactivation or removal of the fluorophores, and then usually it will be in where you have in one cycle 3 to 4 fluorophores that are stained simultaneously. Then you remove them, and then you stain again with a different antibodies with the same 3 or 4 fluorophores. And that way you can reach up to 50 or 100 markers if you do it, for example, 25 times this cycle. It of course takes a longer time to visualize these slides, but the data that you can get is a lot more deep.
Here on the left we can see an H&E image, and on the right a multiplex image, and as we zoom in, we can identify in the H&E several areas, and for example the tumor cells. And around them you can identify a few other cells, maybe some stromal cells, lymphocytes, but we're really limited in the amount of information that we can get from the H&E. Once we start turning on markers in the multiplex imaging, you can see we turned on in red pancytokeratin, and in blue you can see CD20 for B cells. You can really start to see the subtypes and the architecture of the tissue. Here, for example, you can see that we have several B cells aggregate, and also we can see that around the tumor cells we have smooth methyl-actin, which is in green, which can color myofibroblast. Now, if you want to identify whether one of these B cell aggregates TLS, we can turn on CD21. Indeed, we can see in the top right corner a B cell aggregate which is also positive for CD21, which is a marker for follicular dendritic cells, and this is the TLS. In yellow, you can see CD38, and we can see which is a marker for plasma cells, and we can see that this tumor is surrounded by a lot of plasma cells as well as B cell aggregates. And once we zoom out, you can really see the different architectures and zones within the tumor and the amount of information that we can get from multiplex imaging.
So once we have the raw data, which is the multiplex image, what would we do with it? How can we analyze it? And there's actually a pretty standard pipeline for analyzing these images. So it first starts with cell identification. So we do cell detection and segmentation. Once we have the cells, we quantify for each marker its expression within the segmented cells. So we have the marker expression quantification. And once we have that, we can perform cell typing. From the cell typing, we can do neighbor identification, which I will not go into in this lecture. And we can also add this data with the phenotypic marker quantification. And we can use all of these inputs to build our spatial features and to perform a spatial analysis and to correlate it with outcome prediction. And in this lecture, we'll focus mostly on how deep learning can enhance the stages of marker expression quantification and cell typing and phenotypic marker quantification. So what are the current methods to do this? So most methods rely on quantification of mean marker intensity within segmented cells. So I have a cell segmentation. I just do an average of all the pixel intensities within the segment. And that's what I use for cell typing. And once I have this signal for each cell and for each marker, I can do either manual gating, which is decide, okay, above this threshold, this cell is either positive or negative for this marker, or I can do clustering in order to do kind of an unsupervised analysis where usually what I will do over clustering. So for example, define a number of clusters of 150, and then manually I will merge clusters what looks biologically relevant to me, and I will assign them a certain cell type. And these methods have several limitations. First of all, they are laborious and user dependent. So to do that for a high-plex data of 50 markers, setting a threshold for each marker, and then going to another slide and making sure that it also works on the other slide, and then going back to your slide after you corrected it, it's a very laborious and long process. And also, for example, in clustering, the manual merging of clusters is also very user dependent. And another problem is they're really inaccurate and not robust between batches because of several problems that we have in this method. So first of all, we have segmentation errors. Here, for example, you can see an example of a cell which is positive for both CD20 and CD3, which of course, these are two separate cells. One is a B cell, one is a T cell. There's also some background noise that can affect the mean intensity. There's signal spillover between cells. And there's also cells which have cellular projections, for example, macrophages, which can then go into the segments of their neighboring cells and interfere with the cell typing. And another major problem is that these methods are not transferable between projects and slide batches. So let's say you did it once on a slide batch, you did it for several months, you get a new batch, your method is not guaranteed to work. And if you get a separate panel, you have to do it all over again. So how can we use deep learning to solve these problems? So convolutional neural networks really revolutionized computer vision and leading to significant advancements in image classification, object detection, and image understanding tasks. And their main advantage in our case is that they can really learn the spatial context of protein expression and the protein expression pattern. Because if you think about it, usually a nuclear protein which is nuclear expressed will look pretty much the same with different proteins, same for membranous or cytoplasmic. So if you expose it to a wide variety of proteins, it usually can learn the most of the protein expression patterns. And here we can see an example for that. So as you can see here, we have pancytokeratin in red, which is a tumor marker. We have CD68 in orange, which is a macrophage marker. CD4 in blue, which can be positive both in macrophages and is also a CD4 T cell marker. We have FOXP3, which is a regulatory T cell marker, and CD8. And you can see that the signal, it's pretty hard to separate it. So if you think about cell segmentation, for example, for this cell, there's also a vast area which is positive for pancytokeratin. And for here, for example, we can see that there is some unspecific stain of pancytokeratin. We can see by eye that it not relates to a specific cell. And we can see that the deep learning really learns to overcome this limitation and to accurately quantify all of the cells in this example. Last year, we published work in bioRxiv that presents our deep learning pipeline for cell typing and phenotypic marker quantification. And what we did is we developed a deep learning-based framework for both cell typing and phenotypic marker quantification, which really was aimed to overcome all of the problems with mean intensity within segmented cells. And we aimed it so it'd be accurate, robust, and generalizable across different proteins, tumor indications, and imaging modalities, and thus enabling analysis which is reproducible, high throughput across different batches. And as you can see here, we have actually two deep learning components that we trained. So once we do the cell detection and segmentation, And that's the big difference between us and the mean intensity based methods. What we do is we crop, we don't use the cell segmentation. We crop a tile around the cell centroid, and then we can either take the multi-channel tiles and insert them into our deep learning multi-channel multi-class classifier, which can look at several proteins in the same time and decide on the cell type. And we can also decompose this multi-channel tile to single channel tiles, and then our deep learning single channel binary classifier for each single channel can assign it either a positive or a negative positive prediction probability. So in order to check the performance of our analysis pipeline, we used the publicly available colorectal cancer codex dataset that had 140 tissue cores from 35 colorectal patients. And using the markers that were available in this panel, so there were 16 lineage markers, which are specific for certain cell types, and we were able to classify 12 cell subpopulations using this panel. And we can also had 14 cell state or phenotypic markers, which are associated to the immune function of the cells that we also quantified. So we need to separate between the lineage markers, which help us to identify cell types, and between the phenotypic markers, which help us for each cell type to identify its phenotypic state. For example, a CD8 T cell can be either positive for granzyme B, which makes him an activated T cell, or it can be positive, for example, for PD1 and LAG3, which will classify him as a terminally exhausted T cell, which usually does not function within the tumor and does not kill tumor cells. So in terms of the performance, we use over 7,000 annotations, cell annotations to train a deep learning multi-channel classifier, and we assess its performance on 1,700 cell annotations in 14 test tissue cores that the model was not trained upon. And what we saw is actually we get very good accuracy using this method. So we reached 92% accuracy. And again, this is the data that was published in a Cell paper, and they did clustering-based cell typing. And once we compare that to manual annotation, which is the gold standard in multiplex imaging to define cells, we saw that their accuracy was only 64%. And I know it's very hard to see here, but we got very good accuracy in all of the cell types, where in the clustering-based methods, there were some cell types that had really bad performance, such as rare cells like dendritic cells. And I think from this slide, we can learn two important things. One, that deep learning is a highly accurate method for cell typing, a lot more accurate than current used methods such as clustering-based. And second, it's really important to report your accuracy metrics when you do multiplex imaging analysis. Because if you don't know the quality of your cell typing and if your phenotypic marker expression, you might insert a lot of noise into your spatial analysis. And then you won't be able to leverage all this amazing data to get really impactful biological insights. So this is great accuracy, works great, but what are the limitations? So there are two main ones. One, it does not solve us the problem of the phenotypic markers, which each cell can be either positive or negative. And if we start to try to annotate all the possible combinations, it will be very rigorous, time-consuming, and not effective. And second, it's not transferable between projects. So let's say we have now a new panel, a new set of markers. I need to re-annotate all the cells and retrain the deep learning model. In order to solve these two problems, we developed a deep learning single channel classifier, which was trained on more than 90,000 single channel tiles with positive or negative annotations. So what happens here is we take the multi-channel tile, we deconstruct it to single channel tiles, and then we ask our data team to annotate each single channel tile as either positive or negative. And the model does not know from which marker the tile came from. It doesn't know if it's a tumor cell, or it's a lymphocyte, or if it's a macrophage. You just receive a tile and the label of either positive or negative. And that's how we trained our model. And we train it on a wide variety of markers that are expressed both in tumor cells, lymphocyte, macrophages, which are both has nuclear expression or membranous or cytoplasmic. And that's how it really learned to recognize different protein expression patterns. So the first thing that we did is check its performance in the same data set that it was trained on. And we saw excellent performance in more than 20,000 signal channel notations, including in 14 unseen markers. But as you can see here, here we can see the positive and negative distributions in each markers. In the markers that in the phenotypic markers it was not trained upon. And you can see that there is some variation in the positive probability. And that is mostly due to different signal to noise ratio, which each antibody has. So in some cases, it's a little bit more hard for the model to decide whether it's positive or negative. And what we did in order to overcome this problem is we did per channel threshold optimization. Now the question is, how many annotations do we need in order to optimize this threshold? And so what we saw here in the black, we have the 0.5 threshold, so a set threshold for all the channels. And in gray, what we have here is the performance after we did some threshold optimization. And what you can see that in a lot of channels, it really did not matter, the threshold optimization. But there are some channels, especially those with the lower positive probabilities, which benefited from this threshold optimization. And once we did threshold optimization, again, of course, we did it on the threshold optimization on test slides. We validated the performance. We did the threshold optimization in training slides, and we validated it in test slides, which were not seen by the model. And then we asked, how many annotations do we need actually to do this optimization? And it turns out that we only need 20 annotations per channel, and we can get a vast improvement in our results. So instead of now training a deep learning model with thousands of annotations, let's say you have a new panel of 40 markers, and let's say it's something 5 of those we need threshold optimization. We can do 100 annotation in total for the whole cohort, and that's it. We can get very good results. But this was on data that the model was trained on. The question is, so first of all, it's in this setting, it's very accurate in detection of protein positivity, even in unseen markers. And then the next question we asked, how would it work in a different data set, a different imaging technology, different markers? Would it work still the same? So first of all, here we can see examples of the prediction and how accurate it is. So for example, here we used NK TPAs to identify tumor cells and Ki67 to identify proliferating tumor cells. And we were very good in identifying also plasma cells, which are positive for VISTA, which is an inhibitory checkpoint, and also B cells, which are positive for MHC class 2, for example. So now the next question that we wanted to ask is, can we leverage these single channel predictions, right? Because now we mostly did, is it positive or negative for each channel? For doing cell typing, we needed information from several different channels. So can we leverage the single channel predictions to do cell typing by this method? And what we did is usually the number of cells within a tissue are pretty defined, also by the markers that we have and also by our past biological experience. So for example, CD8 T cell is defined by its positivity for CD3 and CD8 and negativity for CD4, tumor markers, macrophage markers, and so forth. So we can say in advance what is the expected marker expression that we want to find in the tissue. And then we can define for each cell type what is the expected marker expression, and we can use the single channel predictions and combine the prediction from different channels in order to do the cell typing. And we saw that we got very good results by using this method. The results that were comparable to the multi-channel, multi-class classifier. But to do that, we only need 100 annotation, not thousands of annotations. And then, so we now know we can utilize non-protein expression within cell classes, and we can reach really highly accurate cell typing from single channel predictions. And then we ask whether it will work in an unseen data. So what we did is we stained 43 melanoma slides, so a different tumor indication. Our model was trained on colorectal cancer patients that were stained for 6 proteins with a PhenoImager, which was previously known as Vectra Polaris, to validate the single channel classifier performance in unseen data. So we have different markers, different imaging modality, and different tumor indication. And then we run our CRC codex trained binary classifier, and we only added 20 annotations for the whole dataset to do threshold optimization in one channel. And we compared its performance against a multi-class classifier, which was trained on more than 10,000 annotations, and cell typing by manual gating. So this is how the data looks like. We can see in white, these are the melanoma cells, which are positive for SOX10, and this is an example which is highly infiltrated by immune cells. We can see in yellow the CD8 T cells, in green CD4, which is the marker for CD4 T cells. In red, we have the FOXP3 for the regulatory T cells, and in orange, we have the CD68 for macrophages. So how did we do? So if we compare ourselves to manual thresholding, we see that in almost all of the cell types, we had a much better accuracy. And again, all we did was to add 20 annotations. Manual thresholding is a pretty rigorous process when you have to go between slides, setting a threshold for each slide. So, and 20 annotations you can do in half an hour, something like that. So when we looked at the F1 scores, comparing between manual thresholding in black in our deep learning binary classifier with the 20 annotations for threshold optimization, we can see that in all the channels we had much better performance. And even when we compared ourselves to the multi-channel that was trained on more than 10,000 annotations, we can see that the binary classifier here in the dark blue reached really similar results as the multi-channel classifier. So from that, we can see that the single-channel deep learning-based classifier is highly accurate and robust across different antibodies, tumor indications, and imaging techniques. I would also add that we do very rigorous normalization of the tiles before we input them into our deep learning models, and that is something that is also very helpful for us to be robust across different imaging modalities. We are able to handle both 8-bit or 16-bit images and so forth. So to summarize, spatial biology is really gaining traction as crucial modality to understand disease, advance precision medicine, and enhance drug development. It is especially relevant in the immunotherapy setting, where we know that the immune cell interaction with each other with the tumor cells is very important, and where there's a growing complexity in the immunotherapies. So right now, we're talking about combination of immunotherapies. So usually each patient will get its PD-1 blockade. And then the question is, which other immune checkpoint we can block to really enhance the response rates of these patients? Because it depends on the cancer indication, but usually the response rates are between 15 to 50%, depends on the modality. And with if we use multiplex imaging to really classify all the different immune checkpoints, we might be able to really do precision medicine and to give each patient the drug that it will most help him. So multiplex imaging allows for simultaneous visualization of multiple protein markers on the same slide, enabling us to accurately define cell subtypes and activation states. And current multiplex imaging analysis methods are not sufficient because they have low accuracy, they lack robustness, and they are very laborious and user dependent. And that's why we developed our deep learning based pipeline for cell typing and phenotypic marker quantification, which is highly accurate and robust across different proteins, tumor indication, and imaging modalities. And that's what really allows us to scale up our imaging analysis capabilities. So in previously, if you had a new panel, 40 markers, usually the analysis will take several months. We can deliver results with less than 1 month usually. And also, that's really what helps us, the robustness across different batches, and the fact that we can give answers really fast to new batches, it's something that we emphasize in order to be able to participate in clinical trials and help with patient selection. So I want to thank all the team at Nuclei, the data team, the algo team, the research team, platform team, software team, all different teams that contributed both to the development of the analysis pipeline and our viewer, and also to our collaborators both in Sheba and the Weizmann Institute. Any questions? The question was if we can train on one channel. So why do we need multiplex imaging? Maybe the deep learning model learn other features of the cell and can predict different channels. So first, it's a very good question. We use a wide variety of channels to train our deep learning classifier, and that's really important because, again, there are not a lot of protein expression patterns. You have nuclear, cytoplasmic, membranous, and then you have also different cells which are in different sizes. So you need like a nice batch from all of these to train the binary classifier. And then it can predict unseen channels because it already saw the marker expression before that. But I will say that it's definitely interesting to try for now. For example, now we can try to leverage our knowledge from multiplex imaging and to try and predict, for example, marker prediction in H&E or different cellular subtypes in H&E. Maybe we can do better identification of H&E cells using our deep learning models, or ground truth, which is based on our deep learning models. So the question was how much the model is influenced by different labs, which has different settings for the imaging, and how robust is it really a one-stop shop for all, or do we need to do fine-tuning for each lab? So I would say, first of all, we're not magicians. So if you have a really bad marker, it's not so much about between lab variation. It's more about if you have really bad marker with really bad signal-to-noise ratio, we probably can't save it, but also the other methods will definitely wouldn't be able to handle it. But we really emphasize developing it as a one-stop shop, actually. So as I showed here, we trained it on one data set that was conducted in an academic center, and then we tried it on a data set that we stained with a CRO in a different imaging technology, different tumor indication. The slides came from a different hospital, so a lot of variation, and it worked really well. Again, we needed in one channel to add 20 annotations, and that was it, basically. So yeah, so it's really robust across different imaging modalities, and I think the way we work is we train it to not be sensitive to intensity variation, and the mean intensity-based methods are very sensitive to intensity variation.
So Ettai, I've got a couple questions.
Yeah, of course.
To throw out there. So, obviously one of the big issues is garbage in, garbage out. We're adding really complex things here. So we're always, I think, under the assumption that we're going to have a good stain that's been validated so that it's marking what we expect. In your experience in taking different inputs from, different multiplexes, just walk through the process. How easy is this? How much time does it actually take? Mm-hmm. If you were to get a new 20-plex from a pharma that it's going to have all the basic markers identifying the cell types and maybe one or two others. What would the process look like for us?
Sure. So There's definitely variation in the quality of data between labs, and definitely there's importance to how you validate your panel. When we trained our model, we of course QC'd all different channels, made sure to input only channels that passed QC. And to your question, Ken, it really depends on the vendor and who is doing the stain and how it did this validation. If you do a very rigorous validation and you use IHC-based ground truth, do single stains to validate your panel, then you do a validation phase for all the panel together, and you make sure in different tissues that you have great signal, then yes, it will make the job a lot easier for us. But during the process of the analysis, especially when we get a new panel, we'll definitely have a panel calibration phase, which go over the channels and make sure that all of the channels pass QC. And if not, we can exclude from the analysis certain channels.
So I'd also like to ask you to elaborate on the annotations in general. So one of the big limitations to deep learning has been annotations off in H&E, which is pretty laborious, a big data team. You got to get people to agree on what individual cells are. And what advantages do you see now being able to use these classifiers to go back and train H&E slides? Can we self-annotate now? How will that work?
Yeah, so that's a great question. I will split my answer to two parts. First, about annotators agreement. So usually in H&E, we know the agreement rate is around 80%. We were actually surprised to see that even though it's a lot more complex data, the agreement rate between annotators and multiplex imaging is a lot higher. It's around 95%. And that's because we have much more validated ground truth here. We can actually know what the cell is by the composition of its markers. And one of the projects that we're working on now is really how can we leverage multiplex-based predictions, because from a certain data set, we can now get millions of annotations using multiplex-based predictions. And then if we have H&E on the same slide, then we can transfer these predictions and use them as ground truth in order to train our model. And what we see is that the predictions of the multiplex images are comparable to annotators in terms of the accuracy, but we can use a lot more data, which can be so again, we run analysis, we can get millions of annotations for a data set.
There's one more question from the audience. When you combine, you do it on a final output or the embeddings?
So when we combine, usually the cell typing is done on the positive prediction probability. So so on the final output of the binary classifier. So usually we get a probability between 0 to 1 for each cell in each channel, and we use this data in several methods to get the cell type. I would just want to add one more to the line to the question that Ken asked before, is that we can also try to leverage the multiplex-based truth in cells which are hard to annotate. For example, plasma cells or macrophages are pretty hard to identify only from H&E, and we here have a much better ground truth for them, and then we can try and use this prediction to identify more granular cell types from the H&E. Yeah, and we are now open to research collaborations. So if you have an interesting multiplex imaging data that you have some difficulties with the analysis, we will be happy to help. And we open our platform for research collaborations. So feel free to contact us. And I will also want to take this chance and present our next lecture in Nuclear Academy, which will be given by Dr. Ken Bloom, our head of pathology, who will talk about AI-driven spatial biology as the next NGS. So thank you everyone for participating, and see you in the next lecture.